So good afternoon, everyone. I'd like to welcome you to Monash Advanced Imaging Seminar. This afternoon, our speaker is Dr. Robert Prevedel from the EMBL in Heidelberg, where he is the group leader in the Cell Biology and Biophysics unit. Dr. Prevedel and his group work on innovative new optical tools and methods that allow observation of cellular processes and dynamics in living organisms in non-invasive ways. Robert holds a PhD in experimental physics, during which he developed new approaches for optical quantum computing, and has now turned that wonderful knowledge to innovative functional neuroimaging in small model organisms. So thank you very much for agreeing to present your work to us today from early morning in Germany. And over to you, Robert. Thank you very much.
Yeah, thank you very much, Stephen. I mean, first of all, for the really nice introduction, but also for sort of organizing this event and inviting me to speak. So while I try to share my screen, I also wanted to say that I really like Australia quite a lot. As I was just mentioning to Steve earlier, I actually did part of my master thesis in Brisbane, still back then working on quantum physics and quantum computing. So it's somehow nice to be at least virtually back in Australia. So today I really would like to tell you about a little bit what my research lab at the EMBL in Heidelberg here in Germany is doing in respect to developing new optical imaging technologies. And so I wanted to start out basically with a nice quote from Sidney Brenner that you might have heard at any one point, which is, "Progress in science depends on new techniques, new discoveries, new ideas," probably in that order. And so I think it's a very beautiful quote that really highlights how important technologies and also the development of technologies are as a prerequisite for doing excellent research in the life sciences. And this is because only with new technologies and in this case, new microscopes, biologists and the like are able to ask new questions or even think about sort of new research that was previously impossible in terms of technologies. But I think, being here in a microscopy imaging seminar series, I probably don't have to really tell you how much, microscopy is enabling various research in the life sciences. We often say here at EMBL, seeing is believing, which goes back to a very successful conference series that we are holding here in the area of bioimaging. And so in this respect, my lab develops various sorts of microscopes in very close collaboration with different labs, both within EMBL, but also outside EMBL. And so we have many areas of expertise in this respect. And we develop microscopes for various applications, be that in cell biology, developmental biology, or neuroscience. Let me quickly bring up my laser pointer, which I hope you can see now. And so, this microscopes are really quintessential and very powerful tools because they allow us to, visualize molecular events, at very high resolution inside cells, and thereby also help us to come to a mechanistic understanding of how this complex entities and then also entire living things, how they operate. So light microscopy, as I mentioned, is a very powerful tool. And it has allowed us, especially with the combination of fluorescent proteins and dyes, the visualization of a lot of interesting biological events. As you can see on the very right, these are sort of cells, that are on a dish. Or here there's a zebrafish brain, where various sort of cellular structures are being marked. And, we all know this type of images. I hope you're also similarly captivated by the beauty of those images. But so when I transitioned into the field of microscopy or bioimaging coming from quantum physics, I also very quickly realized that whenever we were looking at these beautiful images, they were beautiful because, we were looking at individual cells in a Petri dish, or we were looking at, in a way, transparent organisms such as the zebrafish. So whenever the samples are either small, transparent, and still you're not moving around too much, then we get these beautiful images. But certainly once we try to go to, more complex situations where, the tissue or the cells are all very dynamic, where the overall, tissue, for example, is highly scattering, then things become a lot more difficult. And so that I drew as a big inspiration and motivation for my work. And so I set up a lab at EMBL that sort of really aims to address the various challenges there are in bioimaging from a technical side, but always making really sure that these microscopes can very quickly be really applied to biological problems. And so to give you a little bit of an overview of what my lab does, I brought you this slide here on the left. You can see one area in our lab which really deals with trying to image as fast as possible volumetrically within various biological systems. So for example, we do a lot of fast volumetric imaging, for example, in various living systems, such as here you can see C. elegans neurons or neural activity in the mouse. But we, for example, also brought light field microscopy to the area of biological imaging. And I'll talk about this in just a minute. A second big direction in my lab is to try to address the problem of how can you image really deep into highly scattering biological samples. And for this, we sort of use two approaches. One of them is so-called 3-photon microscopy. The other one is a sort of a merge or a hybrid between optics and acoustics. So where we use light to excite sound inside the material. And this is, of course, known as photoacoustics. And last but not least, we've also worked quite heavily more recently on a new research line, which is called Brillouin microscopy, which in a very elegant way exploits actually the scattering that happens of light within biological material in our case. And that allows us to obtain mechanical information of our samples. So whether the cells or tissues are very stiff or soft, or whether the fluids inside are being viscous or not. So I'll sort of highlight various things in these three areas in the next 30 minutes or so. And I wanted to start out by basically talking about high-speed bioimaging. And so here, one of the always present and recurring challenges that we face in biology is the fact that we often want to capture and visualize both subcellular events, like here you can see this mouse embryo and how it's performing cell division at very early stages. But at the same time, we also want to capture tissue-wide dynamics. Here you can see a Drosophila embryo. And all of this, of course, at the necessary spatial as well as temporal resolution. And both of these can span many orders of magnitudes. So, of course, this makes it very challenging for us microscopists to sort of keep up with this sort of requirements. And it mostly relates to the fact that in the most simple approach, microscopes are often point scanning microscopes. So we illuminate a single point to excite the fluorescence. And this point is being, of course, moved around in 2 and eventually 3 dimensions. There's, of course, other ingenious work over the past years, for example, light sheet microscopes that I hope you have at least heard of, where this problem of 3-dimensional scanning is reduced to just 1 dimension because one can also illuminate a full single plane. But still, eventually there's always this trade-off in microscopy between, parameters such as speed, resolution, or field of view. So if you sort of want to really address this in an elegant manner, we really need new conceptual approaches. And this is something that kept me very busy already as a postdoc before I was even starting my lab here at EMBL. And it brought me to the concept of so-called light field microscopy. And so here, light fields are basically just, mathematical descriptions of the entire field of light and how it is propagating in space. And if you think of this as just like in principle photons that are moving on straight trajectories, so we are neglecting scattering here for a moment, then the description of such light fields can basically be broken down to 4 dimensions. So you basically need sort of a point of origin that can be described by 2 parameters, and then sort of the direction or angle under which the light is being propagating. And this can also then be described by basically 2 parameters. So you have a location plus basically an angle. And that allows you to fully describe basically these light fields. Now the, of course, interesting question then to ask is, since these light fields contain all the 3D information of a particular scene that you'd like to capture, how can you actually record a 4-dimensional light field if, the normal cameras that we're all using only have 2 dimensions? And so how to do this conceptually can be sort of well understood by considering this type of gimmick cameras where you actually have different lenses and they all sort of look at the scene at a slightly different angle, as you can see here in the selfie. So it's a little bit like stereoscopic vision, just a little bit more multiplexed. And that, of course, allows you to somehow synthesize 3-dimensional volumes. So we picked up this concept and idea that was proposed actually many, many years ago by a group at Stanford, but that always had a challenge to be actually applied to microscopy. And so we took up this challenge and we designed, in a way, special microlens arrays, they're called, that sort of extend a little bit this concept I was describing before from just a few lenses to thousands of lenses. And by putting such a microlens array into the detection port of a simple epifluorescence microscope, one is able to then computationally synthesize full 3-dimensional information. And without now going into too much detail, I just want to show you how it sort of actually allows you then to look at very interesting biological events. And here you can see the calcium activity or brain activity of an entire zebrafish larvae brain, which spans more than about 700 microns in 2 dimensions and about 150 microns in the Z dimension. And the beautiful thing about light field microscopy now is that the volumetric data here is being recorded basically at the rate of the camera. And as you all know, modern cameras can be operated actually at many, many hertz. In this case, we only run the camera at 20 hertz. It could have gone much faster. But I think it hopefully allows you to still see that within the fraction of a second, here we have complete entire volumetric information that with a confocal would probably take many minutes to acquire. And so if you're a little bit captivated now by this field of light field microscopy, I mean, over the years, this has been then established in various model organisms, both by my lab, but many, many other labs. Some of them are being mentioned here in these papers. And there's also a website where we describe how to turn a simple epifluorescence microscope into a light field microscope. It's actually, from a hardware perspective, is very straightforward. All you need is basically a microlens array and some additional optics that you can append to most side ports of commercial microscopes. So that all sounds really great. But then, of course, getting more and more into the meta, I also realized that there's a couple of drawbacks that are inherent to the way that we have done light field microscopy that sort of also really compromises some of the performance. And so one of the drawbacks is that the optical resolution that you get from synthesizing this 3-dimensional information and reconstructing it computationally on a computer is particularly bad in the Z direction. I mean, this is not unusual in microscopy. We all know the lateral resolution is usually much better than the axial one, but this is especially bad in light field microscopy. And in fact, stems back to this trade-off that we need to go into if we want to obtain 4-dimensional information on a 2-dimensional sensor. So both recording, spatial information as well as angular information. That's basically where it all comes from. A second particular drawback is the fact that the volume synthesis is being done computationally. And without, again, going into much detail, it leads to the fact that around the focal plane, there's a so-called artifact plane, where this reconstruction of a high information structures is not really possible. And the third drawback that we all realized is that if we actually only interested in sort of reconstructing a small volume out of a bigger chunk of, let's say, tissue, then of course, so-called out-of-focus fluorescence that emanates from above or below this region of interest sort of bleeds into, of course, our sensor. So the sensor still records all of that fluorescence, but cannot sufficiently trace it back to this volume of interest. And that sort of basically creates a lot of background, as you would also expect it from other microscopy approaches. So when I then set up my lab at EMBL many years ago, I actually wanted to chip away at some of these drawbacks in order to make light field microscopy more suitable to some of the more challenging bioimaging tasks. And so we've done that by basically combining the best of two worlds, namely light field microscopy and light sheet microscopy, which I hope you've all heard of. And by combining this, it allowed us to, for example, reduce the out of volume fluorescence by basically introducing an orthogonal light sheet into our sample, thereby only illuminating the volume of interest. So just to be aware, in light sheet microscopy, we normally form a very thin sheet of light, typically just a few couple microns. Here, this is a super thick light sheet in a sense that we are sort of usually designing this to span around 100 or even 200 or more micrometers in the Z direction. The second approach we took in order to chip away of some of the limitations was to introduce something else that has been developed in the field of light sheet microscopy, namely multi-view detection. And this goes back to some papers that were also performed earlier at the EMBL, where researchers have started putting various lenses in different configurations, basically allowing you to look at the sample from opposing or 90-degree different sides. And this helps you in terms of resolution, because as we all know, the point spread function is highly asymmetric, right? It's always longer in one direction than the other. But then if you sort of combine two views from an orthogonal direction and you sort of fuse the two together, you end up essentially with an isotropic point spread function. So we took these two ideas, the orthogonal illumination plus the orthogonal detection, and we built a microscope that we called ISO-LFM, which basically is this like maybe somehow complicatedly looking arrangement of three objective lenses that are all 90-degree to one another. But then this really allowed us to reconstruct biological samples at much higher accuracy. So here you can see a medaka fish. It was mounted basically in this sort of arrangement where the illumination comes here from the top, from the front, and then we had two orthogonal detection objectives that would sort of take up the fluorescence and image the light fields. And sort of by doing that, you can hopefully appreciate here on the top right, you can see the reconstruction of a beating medaka fish heart. And on the top, you can see the light field only reconstructed from a single objective. Excuse me. And on the bottom, you can see how it looks like if you merge the two views together computationally. And especially here in this axial direction with the zoom-ins, you can really see that with the dual view, you can now resolve individual nuclei here in the myocardium, while with a single view, it becomes, difficult at best, but sometimes even impossible to track these individual cellular nuclei. And another aspect I want to really highlight here is that, with these light field microscopes, we can really look at large samples here around 200 microns in each direction at basically the frame rate of our camera. And in this case, we ran it already much faster, about 150 Hertz. So we really have a volumetric imaging at a super high frame rate. Furthermore, we could also sort of actually look at some interesting biology. Here you can see, again, the reconstruction of a medaka fish heart, but in this case, we are not labeling the myocardium or the outline, but we actually label the red blood cells that are flowing through it. And as you can see with the single view, sometimes it becomes impossible to really tell and track the individual particles. But with the dual view, they all become nice punctures, and we can actually track them. And what was interesting to see is if you notice or if you follow some of these cells closely here, some of these red blood cells actually stay in the heart chambers for a couple of beating cycles. So it's not like that all the blood is being flushed through in one go, but actually some of them end up sticking around the heart chambers before they eventually being flushed out. Okay, so this is a nice demonstration of how you can actually improve the resolution of light field microscopy. I already mentioned that light field microscopy is able to image very fast, basically at the frame rate of camera. Now, of course, it shifts sort of the difficulty of the whole technique basically to the computational side. Because, of course, we can require a lot of data very fast in the camera, but each of those frames need to be reconstructed in order to give rise to these volumes. And so far, I haven't talked much about it, but I wanted to do so now in the next 1 or 2 slides, because it's also still one of the major challenges in the field. So we are basically moving all the difficulty from acquiring the data to actually reconstructing the data. And this is time consuming normally, because the way we go from a light field recording to a 3-dimensional volume is by a so-called deconvolution process. And deconvolution processes, they typically run iteratively, and we need many iterations in order to come up with a high resolution reconstruction of our samples. And this typically takes quite a lot of time. And while you can, of course, parallelize this process on, for example, a CPU cluster, it will still take you many hours to maybe even a few days to reconstruct long time series. Because, of course, if you are acquiring 150 of these frames a second and you're recording maybe over a couple minutes, then you can easily understand how much data is eventually being reconstructed. So we also wanted to work on that end, or on that challenge. And for this, we teamed up with an artificial intelligence group at EMBL, the lab of Anna Kreshuk. And she helped us design a neural network that basically learns how to do the deconvolution. So then instead of doing an iterative deconvolution, the neural network can then be inferred basically in a few tens to maybe hundreds of milliseconds. And that allows you very quickly then to reconstruct this volume, so to go from the light field to the volume within just maybe a few tens of milliseconds, thereby effectively speeding up the whole reconstruction by 3 orders of magnitude. And here again, you can see another visualization of the speeding with darker HARP. You can basically see that here in magenta, the reconstruction quality is almost unchanged from the one that I showed you earlier. But, it is, of course, reconstructing much faster. All right, so with this, I wanted to basically transition to one of the other areas in the lab that does not maybe concerned with very fast imaging, but with sort of trying to image very deep, especially into highly scattering tissues. And so before sort of going more into the technical details here, I wanted to really say that all of the inspiration here again comes from the area of neuroscience, in which we really have this urge to understand how the brain works. And brain matter, as it turns out, at least in most higher animals, is quite scattering and opaque. And especially here in the case of the mouse brain, it again creates a lot of challenges for us microscopists. Again, it's related to different scales, especially the spatial scales, because in the mouse brain, the spatial scales span various orders of magnitude from the whole brain down to individual dendrites and synapses. And so here we really work a lot in the area of multi-photon microscopy, and we try to really push the resolution of this technique in terms of being able to record from larger volumes, a little bit like what I've described before with light field, but also remaining or retaining high spatial resolution. But one of the most difficult areas here is basically to try to image very deep into the brain, as you can see over here. And so this is a little bit, this challenge is a little bit akin to when you maybe drive, I think now it's winter, right, in Australia, when you try to drive home, maybe from work, and it's a foggy evening. You all know how it's difficult to really see ahead on the street. And then if you try to turn up your headlights, you will immediately notice that actually it doesn't improve your ability to really see far. You actually just get blinded by a lot of backscattering light. And of course, the essence here is that this is very similar to the challenge that we face in microscopy when we try to image very deep into highly scattering optical material. And of course, the underlying problem here is light scattering, so the interaction of light with matter, which essentially leads to the fact that photons don't propagate on a straight line anymore, but are basically sort of scattered around and deviate from the straight ballistic path. And so how does that affect, of course, biological imaging? Well, as I said, it's a little bit similar to this headlight situation in a foggy night. And so what we, of course, in neuroscience would like to do is we would like to visualize the whole brain, ideally, of the mouse at very high spatial resolution and with a very good contrast. But the problem is, I mean, this here on the left is a cleared brain slice, right? But then if we would sort of go in here from the top and we try to image deeper into the cortex, at one point we essentially lose the contrast. So we get blinded by a lot of this backscattered and out of focus fluorescence light. And this is, of course, a challenge because neuroscientists want to trace individual neurons and their dendrites. They want to maybe even understand how they sort of connect to various other neurons. And for this, they really need this high contrast, this really good signal-to-noise ratio to really make out these fine cellular protrusions here, the spines, for example. And the challenge here really is to can we sort of retain the high resolution that optical microscopy affords us, but then can we push this to very, very high tissue depths? And to understand this challenge a little bit more, I brought you this graph here on the left that basically plots the effect of attenuation of the light, which is a combination of absorption and scattering, as a function of the wavelength of the light. And what you can see here clearly is that the further you go towards the infrared, the higher the attenuation length becomes. And attenuation length basically means how far we can go into the tissue before significant scattering occurs. And so what you can see here is that normally in multi-photon microscopy, people operate here at around 800 or 900 nanometers to excite green fluorescent proteins. However, if you would move more into the infrared, you can see how the curve here shifts up, which basically means you can penetrate much deeper into the sample. And indeed, there are sort of two, let's say, local optimums in this curve here, one at 1300 and one at 1700 nanometers, that both sort of are able to excite either green or red fluorescence through a so-called 3-photon excitation process. But first of all, I wanted to show you how just moving the wavelength from, let's say, 800-900 nanometers to the infrared really helps you in imaging deeper. Here, this is some data from the Schul lab. This is mouse brain vasculature. And you can see here on the right, it's about 800 nanometers. Here on the left, I think 1300. You can already see how remarkably the imaging depth is being improved here, almost by a factor 2. So, longer wavelength affords deeper imaging or less scattering. But what also helps us tremendously in this area is that if we go to a 3-photon excitation process, let's say at 1300, which we need to excite green fluorescence, we also markedly reduce the out of focus background. And here you can see this strikingly in this example between 3-photon and 2-photon image of the same vascular data here. This is the same slice inside the brain. And you can see how remarkably different the SNR is here. And this is just because in 3-photon microscopy, it's much less likely to excite some out of focus fluorescence, which mostly emanates from the surface here in the brain. Because as we try to image deeper, we of course try to crank up the power, but then at one point, we are exciting actually fluorescence that are further up in the brain, and this sort of contributes to this background signal. Now, it's not just always about reaching deep or achieving high imaging depth, but it's also very important to maintain a very good optical resolution when you try to do this. And this is actually a big challenge in the field, since optical aberrations from both the microscope, as you can see here, but also because of the sample and the heterogeneities inside the brain in this case, really deteriorate the resolution with increasing depth. basically blurs out our point spread function because less and less ballistic light really reaches the actual focus. So ideally, of course, one would like to revert these aberrations or wavefront distortions. And if you were able to do so, you could basically ensure that all the photons hit the exact focus inside the brain. And this can be done by an approach called adaptive optics, which is borrowed in a way from astronomy, where they've developed technologies such as deformable mirrors that allow us to flexibly and arbitrarily change, for example, the wavefront of laser light. And this is basically just because we sort of mend these membranes to the exact shape that we want the wavefronts to obtain. Now, why is all of this important if we want to image inside a mouse brain? Well, remember, you know, very often neuroscientists want to visualize very fine processes here, for example, along the dendrite, the spines, which are only on the order of a micron or even smaller than a micron. Now, if we have a very elongated point spread function, such as you can see here, especially in the Z direction, then the fluorescence that we get from one of these spines is of course very small. But if we're able to use adaptive optics to compress basically the point spread function to the smallest possible size, then of course we can get a lot more fluorescence out of these small structures. So adaptive optics helps us in two ways. First of all, it increases the fluorescence signal just because it sort of puts more energy into a smaller region. But it also makes the image appear a lot sharper because, of course, it also compresses the point spread function, and which is basically the same as optical resolution. And so our goal many years ago was to basically combine these before mentioned techniques in order to chip away at this infamous pyramid of frustration that I think I already alluded to in the beginning, that we all know in microscopy it's very hard to optimize these various parameters without affecting the other. But by introducing new techniques, we can basically circumvent or avoid some of this. For example, we can use 3-photon microscopy to reach much deeper into the brain, and then can use adaptive optics in order to make sure that the resolution is as good as possible. All of this while ensuring that the overall light dosages that we put into the mouse brain are at a level that is not affecting the physiology. And so here on the left now you can see a simple schematic of how a 3-photon adaptive optics microscope looks like. When we set out to design this microscope, we were very diligent in trying to optimize the various parameters that affect fluorescence excitation through a 3-photon process here, and especially the repetition rate and the pulse duration of the laser. This made us choose a very low repetition rate laser here, but with a very high pulse energy at a short temporal duration. And this all assures that the most possible light is being delivered deep into the brain, while still making sure that the pulses are short enough to excite fluorescence through a 3-photon process. Now here on the right, you can see how this sort of looks like in the lab. It's a lot of messy optics, but I think much more impressive than just this assortment of various optical things is actually the data that you can acquire with such a microscope. And so remember, normally with a 2-photon microscope, typically one can reach very high resolution data up to a depth of around maybe 500-600 micrometers, which you can see here. But now with the 3-photon microscope, we can more than double the imaging depth here, and not only record through the entire cortex of the mouse, but also go through the corpus callosum and into the hippocampus. Now I can also show you how adaptive optics really also visually improves the quality and also resolution of the images. Here on the top is an image taken without adaptive optics. Here with adaptive optics, you can immediately see a lot more fine details, which become really visible once you zoom in. And here this is a single dendrite. And you can see here same parameters for the two acquisitions. On the top, you can barely make out the dendrite itself, but certainly no spines. But here on the bottom, you can see all these interesting small protrusions. But we can not only do this in the cortex, we can also go really past the corpus callosum and into the hippocampus, the subcortical structure that is very important in neuroscience. And you can see here that across soma, for example, with adaptive optics, we get a threefold improvement in the actual signal that we can acquire from these neurons. And this sort of improvement is even higher if we go to very small structures such as dendrites, where the improvement can be even 8 or 10-fold. And this is maybe not surprising, because if you remember the sketch that I showed a couple slides back, especially when the structures are smaller than the point spread function, as is the case for the dendrite, but not the soma, especially here, sort of compressing, of course, the point spread function leads to this immense improvement in fluorescence, since it scales with the cube of the power that we put into. And so here's a last demonstration. I'm showing you some data that we acquired fully within the hippocampus of between 900 and 1500 micrometers. And again, you can see markedly how here the dendrites are much, much brighter with adaptive optics. Okay. But then, of course, we always, as I said, we want to apply our tools to really interesting questions in biology. And here for this microscope, we teamed up with a very talented neuroscientist, or neuro-oncologist, I should say, at the University of Heidelberg, who's really interested in studying the process of glioblastoma, so brain cancer invasion, which is well known to happen both in humans as well in the mouse through the so-called corpus callosum layer, so the white matter that you've been already seeing in one of the previous images. So that the corpus callosum fibers here really act as sort of a highway that allows these glioblastoma cells to migrate to various brain regions and infect them, or invade them, I should say. So glioblastoma are really often called a so-called white matter disease. And of course, many people still don't know how exactly the cell migration happens on that level, especially at this deep layer such as the white matter. And for this, you actually need to study the biology in vivo and over time. And therefore, you need non-invasive imaging tools to actually allow you to sort of, in this case, look at the various migration mechanisms that these cells are taking at this high imaging depth. And so this was a beautiful, let's say, application of 3-photon microscopy. And so we started out to actually try to get some data for this. So for this, we are parking our microscope basically at around 1000 micrometers or 1 millimeter inside the brain. This is the area of the corpus callosum. And then here, as you can see on the right, we can record beautiful movies of cell migration. Here on the top, if I go back, these are so-called branching migration behavior. Here in the middle, we see some translocation here of the soma, as indicated by the arrow, or more straightforward locomotion of the cells. And interesting, what we find is that some of these cell migration patterns are actually more prominent in the corpus callosum than in the cortex. So it's been studied a lot, of course, inside the cortex with 2-photon microscopes. But what we now actually found is that the migration patterns are very different inside this corpus callosum layers. Now, something else that we needed to do in terms of this project was we wanted to better understand how these invasion routes of the glioma cells, how they would look like. But for this, we not only need to visualize the cell itself, but also its environment. And the environment in this area, in this case, are both the white matter tracts, but also the blood vessels. But both of those basically give rise to the same third harmonic label-free contrast. So while fluorescence, we can directly detect the third harmonic is composed of both the myelin and the vessels. But of course, they're very different biologically. So we first needed to sort of differentiate them somehow. So for this, we customized some machine learning tools. And it allowed us to basically, distinguish between the myelin tracts in the corpus callosum and the blood vessels, and thereby sort of make this multicolor images where we then could really place the glioma cells inside this environment. All right, so with this, again, maybe switching a little bit of gear, I told you how we image deep with optical techniques. Another quite interesting avenue here is to explore the field of so-called photoacoustics, in which we don't detect fluorescence, but sound. And so this basically goes back to this motivation, if we want to image as deep as possible, at some point, optical techniques, whether that be 2 or 3 photon microscopy, at some point, they bottom out. At some point, there's just all the light will be lost due to scattering. However, acoustic waves, because of the lower wavelength, they are much a higher wavelength, I should say, lower frequency. They are much less scattered and therefore penetrate much, much deeper before being affected by scattering. And we all know this because, if we do an ultrasound, some of our organs, we can see centimeters deep into our own body. And so in between ultrasound and microscopy lies the field of photoacoustics, which is basically this hybrid between light and sound, where we excite sound waves by using laser pulses, but then we detect the sound. And because the sound is being detected acoustically, it allows us to sense these waves from much, much deeper inside our samples. In our case, being optical physicists, we actually use a so-called optical technique to detect the sound waves. I don't go into much details, but these are really custom fabricated small membranes that basically act like as like etalons, and that basically translate any impeding pressure wave, the sound waves, right? These are pressure waves, into an optical interference signal. And we can now then take this and reconstruct, for example, here, blood vasculature inside the mouse at a very high depth, so this is at 5 millimeters, at mesoscopic resolution. And this is really exciting for us because remember, in multi-photon microscopy, we can visualize cellular activity inside the brain, but maybe only towards a millimeter or 2. Certainly not on the whole brain level. On the other hand, there's this microscopic technique such as functional MRI, where you can detect the whole brain, but at super poor spatial and temporal resolution. So really for us, photoacoustic microscopy is sort of a little bit this bridge of the gap, which allows us to go to a higher field of view, but at a much higher spatial and temporal resolution. And here the crux at the moment, I have to say, is not so much maybe the photoacoustic technologies, but also the fact that actually there's no good molecular probes that allow us to sense the cellular activity, in this case, neuronal activity, for example, the calcium. There's fantastic molecular tools for optical microscopy. You might have heard of GCAMs that translate calcium levels into fluorescence, but there's little to no such probes that do the same for photoacoustics, that basically translate calcium levels into acoustic sound generation. And so to basically address this, we teamed up with the group of Claire Deo, a fantastic chemist at EMBL, and Alex in Claire's lab and Nikita from my lab set out to screen a variety of new probes. And they were able to identify a couple of very promising photoacoustic calcium probes that they engineered. And very recently, we basically also start putting them into mouse brains, and we can indeed detect them as very bright signals. Here, for example, we were trying to actively label the hippocampus inside the mouse brain. So this is on bioRxiv, and we are working now more on getting this into a sort of useful tool with which we can actually at one point not just maybe label the hippocampus, but hopefully entire mouse brains. All right, so I believe I have maybe 3-4 minutes left. And so I wanted to tell you during this time about the last pillar in our research line, which is about Brillouin microscopy, this tool that we use to basically sense mechanical properties by using light. And the field of sort of biomechanics I often call the dark matter of modern biology, because we are all used to using fluorescence to mark specific things inside cells and tissues. But how can you label a physical property of a biological system? How can you label, let's say, physical forces or mechanical properties? And especially the latter for us, we find quite interesting, because it's been very challenging to impossible to come up with fluorescent probes that somehow report the mechanical environment inside a living system. Yet mechanical properties are, of course, very important, because it has been, for example, shown that here in this case, stem cells develop to different cell types depending on the substrate on which they were cultured. So if the environment is very stiff, the stem cell will also differentiate to a stiff, for example, bone cell. While it's very soft, and it will differentiate to a neuron. Now, the problem from a technological point of view is that the typical tools that are usually being used to assess cell mechanics have a lot of limitations. They are most of the times so-called contact-based. So either an atomic force microscope or a micropipette is being put in direct contact with the sample and is exerting a force. So you're either pushing on your sample or you're pulling on your sample. And that is, of course, quite invasive and doesn't allow you to go 3D. There's other techniques that are then either have very low resolution or somehow otherwise invasive because it relies on the introduction of foreign matter to the biology. So overall, in the field of mechanobiology, there's a lot of limitations from a technological point of view, with techniques being either invasive or resolution or only limited to the surface, like these two here in the beginning. And so this is why we started to work on Brillouin microscopy, because it's, for me, a super elegant approach that uses light and allows you to basically visualize mechanics in the same way in which we use microscopes to visualize biology. We can, in a way, directly map mechanical properties at high spatial and temporal resolution. And so this technique works by actually exploiting light scattering, as I said. So any, let's say, matter, whether that's biological or not, is typically not at rest, but undergoes tiny fluctuations. These are thermally agitated fluctuations. And this is basically because the matter is not at 0 Kelvin temperature, but let's say at room temperature. And so these small collective vibrations of the atoms inside the matter give rise to so-called acoustic phonons. And these acoustic phonons can scatter light. And if they scatter the light, they are called Brillouin scattered photons. And so if we record only the Brillouin scattered light, so not the Rayleigh, just the normal scattering, but the Brillouin scattering will give rise to a unique spectrum, which you can see here on the right. So at 0 frequencies, the Rayleigh light, this is basically inelastically scattered light, but then a small portion is being elastically scattered so that it exchanges energy. And it gives rise to this Brillouin spectrum. And for both the shift of the spectrum and both the line width, we can basically deduce mechanical properties that are related to stiffness and viscosity parameters. And so these are basically is a technique that is very challenging because we need to really resolve this very tiny frequency shift of the photons. Luckily, there's now advanced optical elements such as these ettalons called vipers that allow us to really have this very high dispersion. And that allows us to basically map the Brillouin spectrum you can see here. So these tiny dots here, this is the Brillouin spectrum, the white lines are the Rayleigh, to basically sort of measure these tiny frequency shifts. And the neat thing is that this is basically a spectrometer, and a spectrometer can be, basically appended to any sort of normal microscope. And so over the last couple of years, we've designed various sorts of light microscopes. Here you can see a commercial body. It's sort of what we've started with in the beginning, and that allowed us to obtain mechanical information on various scales. Here you can see here on a subcellular scale. You can see the nuclear envelope and nucleoli. We can do this on a tissue level here, for example, in a zebrafish. But we can also, for example, image entire embryos. Here, this is a Felucia embryo, for example. So Brillouin microscopy really allows us to capture now not only with fluorescence the molecular information, but with Brillouin also the mechanical information. Here you can see some data from mouse embryo development, or here from Drosophila morphogenesis, where the tissue invaginates, and you can see these tiny changes here in the Brillouin shift that indicate a change in tissue stiffness during this time. So I need to slowly wrap up, but I also wanted to sort of give you a little bit of an idea of how much potential there still is in this field of mechanobiology and the role that Brillouin microscopy can take into it. So we, in our lab, we spend a lot of time on technical development of these Brillouin spectrometers, in making them faster, in making them more sensitive, we've had some nice papers here in the more recent times where we showed different approaches that can allow us to do that. But we also spent a lot of time now more recently working really closely with various biologists, but also clinicians in order to explore the role of mechanics in this environments. For example, we've teamed up with some cancer biologists in Heidelberg, where we could show how basically the environment, but also the cells themselves in tumors are being more stiff. And this eventually sort of might allow us to even in the future to combine these technologies with what's already available in clinics, such as sort of fiber endoscopes, which at one point might even allow clinicians to probe the mechanics of cells and tissues inside even human bodies. And there's actually quite some nice work that's also happening, I think, at the moment in Australia in this direction. All right, so I think with this, I'm more or less at the end. I think I took 45 or 50 minutes. I just wanted to mention that in Heidelberg, we have also very nice environment. I mean, it's not always as sunny and nice as it is in Australia, but sometimes you get this really nice views here up on the hill where EMBL is. And we're always looking for very talented and highly curious people at all various levels. So if you're interested, please don't hesitate to get in touch either via email or by visiting our website or on Twitter. So with this, I'm happy to take any questions. Thank you.
All right, thank you very much, Robert. That was fantastic and fascinating. Let's see, we've got a question here from Juan. Let me allow you to talk, Juan, so you can ask your question.
Hello, can you hear me?
We can.
Yes.
Yeah,
Hi.
cool. Hi. So my question is about the light field. I think it was very cool, very interesting, and I really like the approach. But in my circles, I've seen a lot more buzz around lattice light sheet as a way to do fast imaging and fast volumetric imaging.
Yeah.
But it gives you like a shear in time, which you can, I see most people ignore it, but you avoid it altogether in light field. So I'm wondering like why you think that is, or is it just that I'm not plugged into the right communities?
Yeah, I mean, I think at first, I think maybe the light field and lattice light sheet occupy maybe slightly different areas, inside this pyramid of various performance parameters. So with a lattice light sheet, I think you can get a lot higher spatial resolution. You certainly would not be able to capture an entire volume within just a couple milliseconds. But the most lattice light sheet microscopy is applied to cell biology questions, which are maybe not the right time and space as for light field. I think light field is really interesting for super highly dynamic events, whether that's, for example, neural activity, or as you see, it's always this beating hearts, something that is more dynamic than intracellular motion, let's say.
Yeah, cool, thanks.
Okay, we also have a question from Michael Hickey. Let me just allow you to talk there, Michael.
Can you hear me?
We can.
Yeah.
Okay.
Hi.
I'm a simple man. I understand the concept of 3-photon microscopy, and you get the 3 photons together to excite your fluorochrome. But ultimately, what you then do is that you're exciting the emitted light is the same wavelength as we use in confocal and 2 photons. So, how do you? Doesn't the light coming back out of the tissue still have the same issues affecting it as it does with conventional microscopy, and how do you overcome that?
Yeah, no, it's a good clarifying question, and unfortunately, I don't have time to go into all the sort of detail and background of every technique. No, so the big difference here to realize between confocal and multi-photon microscopy is in a confocal, you're trying to really filter out the ballistic light, in that case of a pinhole. So you're trying to avoid the scattering of the emitted light. But in multi-photon microscopy, you basically ensure that only a single point is excited. then the emitted light, as you said, in the visible is still highly scattered. It gets out of the sample highly scattered. But then it's basically all being detected by one detector. And then sort of we basically put it back into where the focus was, right? So that the detector behind the objective collects all the light, whether that's scattered or not. But
Right.
since we only excited a single point, we don't care about the scattering of the emitted light. Maybe I hope that makes it clear. So we don't care about the scattering of the emitted light. We actually just want to correct the scattering of the light going in to excite, because in multi-photon microscopy, it's about ensuring a tight focus.
Right.
And
So
not
we know what the plane of focus should be.
Exactly.
Sort of, yeah. Okay. Yeah, that makes sense. Thank you.
No, I'm glad I could resolve that. Yeah.
Excellent. Do we have any more questions from the floor? Then I will ask one if I can. On the last topic there, the Brillouin microscopy, what is the limitation? You're showing different things across an embryo at one stage. Is this an average of the mechanical properties across that entire thing, or does it work in the same way that there's a focal point in the middle of that? I don't get that that part.
Yeah, no, it's actually an excellent question. Again, I didn't have enough time to sort of talk about it. So you can think of this as like a confocal microscope. So you're basically, in this case, you're focusing to a single point. You're collecting the emission, making sure it's not scattered, and then you analyze the spectrum of it. You are completely right. So we are basically essentially measuring an average of all the mechanics that are within the point spread function. And that very often can be actually a challenge or a limitation, because, within, let's say, 1-2 microns of our point spread function, there could be various different mechanical constituents inside, right? So that's certainly a little bit of a limitation, because we are essentially averaging over the mechanics. Still, here we're looking at an average of mechanics on the order of maybe 1 micron or 2 microns in space. If you consider any of the other techniques that I think I mentioned in the beginning here, all of those are basically measuring mechanics on much larger scales, right? The tip of an AFM is also a couple of microns, or micropipette is even tens of microns, or optical elastography is again tens to hundreds of microns. So we can't really measure the mechanics of nanoscale, things, but at least of micrometer scale things, and we have to average over those.
Okay, fantastic. So I think in that paper from last year where they were doing pulsed stimulated Brillouin microscopy, where you said you could take the energy down by using the pulse laser. So there's no bonus then in keeping that energy nice and high and trying to have, a larger field of view, a larger depth of penetration, because it's just going to average across that field anyway. Yes.
Yeah, I mean, yeah, I mean, in a sense, of course, we have to obey photo damage limits, and that's a bit of a challenge in always in microscopy, but especially Brillouin, because the signals, the scattering is so weak typically, right? That the first thing that people try to do is, right, they want to crank up the laser power, right, to get more signal out and then be faster or deeper. But that's, of course, in Brillouin microscopy not that easy, because you would need to go to power levels that are quickly becoming damaging. I mean, one way we sort of circumvented this is by, for example, going again more towards the infrared, using higher wavelengths lasers. And that sort of helps. Or, for example, pulsing, as you said, it's a different approach. I didn't talk about this too much here. I didn't have the time. But then you can actually also more effectively generate scattering events inside the sample.
Excellent. Well, thank you for that. I think we're done with questions from our attendees. So let me thank you once again for a fantastic talk, going across a brilliant range of different modalities there. So I wish you a good day, and I hope the others who have viewed this have enjoyed it as much as I had, because it was certainly great and eye-opening. Thank you.
It was a pleasure, yeah. Good day, everyone, and hope to see you at one point.
Okay, cheers. Thank you.
Bye-bye.